So one of the ways we'll measure growth in the laboratory is through turbidimetry using a spectrophotometer, but we'll also use the tried and true plating method, viable plate counts. And so we'll actually be able to combine these as well, the cell growth through turbidimetry and then cell growth through viability assays like plate counting. So with turbidimetry, obviously you have a specimen, and depending on the density of the specimen or the cell density of the specimen, that dictates how much light can proceed through. And obviously there's a collector on the other end that registers that on a spectrophotometer. So more cells equals more scattered light equals less light detected. And therefore there's an inverse relationship between the amount of light gets through and the number of cells that are present in your culture. More cells equal less permeability. And that, when we look at it that way through turbidimetry and viable counts, plate counts, and so forth, we can generate curves based on log viable cells in the case of viable counts, or optical density versus time when we use a spectrophotometer. And so again, here's our line for turbidimetry, and then here's our hypothetical graph for viable count. And either way, depending, regardless of how we do it, bacteria will fall into the characteristic four categories of, or four phases of growth: lag phase, exponential, and stationary phase. And then there's this death phase where things taper off. The lag phase is obviously where the cells are gearing up for their life in that particular medium. And then the exponential phase is when they've sort of figured it out, and then they grow at an even proportional rate, and then eventually they taper off as nutrients dwindle and waste products build up and so forth. And so again, but the interesting aspect of this is with when you do these turbidity readings, you're not really looking at necessarily live and dead cells. You can't distinguish them. So what's happening in these phases is interesting, and something that we tend to like to look at from a physiological perspective, just to kind of get the sense that, is this population really just static, or is there something else going on there? Another way to look at this is, we look at this number of cells per time, and again, we see the clear lag phase and then obviously the exponential phase. And using this approach, we can directly from the y-axis look for where the population of cells doubles, and then extrapolate to our line of best fit in exponential phase. And we can use that information to get the generation time or doubling time, the amount of time it takes for the population to double. Typically, we like to write that. We like to record that information in minutes, and so that leads us to these important equations that we'll be using in the lab portion of the class. The first one is growth rate, and then the other one is doubling time. And so again, forget about the derivation. You can go through this and look at how the equation is derived, but the end result is mu, which is the growth rate, equals the log of the log of the final population minus log initial divided by 0.301 t. So 0.301, the rate constant, and the time between time points. And so again, you can do that by taking this number, so log final minus log initial divided by 0.301 t, again, the time between those, and that information will give you the generation time. So theoretically, any 2 points on this graph, where the line of best fit is the steepest for exponential phase, should give you the growth rate for the organism if you plug the variables into this equation. The doubling time is just doubling time g equals just 1 over the rate. And so again, the simplest way to look at it is if your doubling time is 20 minutes, for example, so that means that the population doubles every 20 minutes, then your growth rate, which is the number of generations per unit time, and usually this is generations per hour. We usually talk about this per hour so we can be more apt to have whole numbers. But if you're and with doubling times, we tend to put these in minutes. So again, if your doubling time is 20 minutes, meaning that the population doubles every 20 minutes, then your growth rate per hour is 3. So it would be, again, 3 generations per hour. This graph represents dioxic growth, which is a variation on this particular theme here. Here you see the bacteria undergo a lag phase where they're gearing up, and then a very smooth exponential phase. Nothing's really changed. The conditions in the medium are consistent. The temperature is consistent. The amount of shaking or no shaking, whatever it is consistent. And so there's no variation. And so the cells grow at an even rate. But in this hypothetical example, growth per time is disrupted. And so you have a lag phase of variable length, and then a log phase, which is interrupted by a second lag phase. So in this dioxic growth curve, the variable in this case is the type of sugar present. And so again, the mixture of sugars of lactose and glucose in the medium cause the microbes to undergo this dioxic type growth. They're always going to go for the simplest to digest, the monosaccharide glucose. They'll eat that up, and then once they've eaten up all the glucose, they'll switch their genetics and upregulate the activity of other enzymes that are capable of breaking up the disaccharide lactose. So that's where the second lag phase comes from, and then they'll have a second log where they eat up the lactose. So this is an example of dioxic growth, and microbes will do this with all kinds of compounds. They don't just gobble everything up at the same time. They're like little kids in that way, where they'll eat the preferred thing first and then move on to the more difficult to consume. And again, I'll get into the mechanism of this later on when we talk about metabolism and so forth. So death phase is not just what it looks like here, just a predictable slow drop off to 0 or whatever. It's not like that. It's a little bit more sophisticated than that. And so is stationary phase, and that's what I was alluding to before. So there's alternative hypotheses for what's happening in death phase, one of which is the VBNC hypothesis, where the cells are alive, but they're dormant. And so I often give the Vibrio fischeri example. But in that particular case, you can take some cells of Vibrio fischeri and put them into seawater and plate the seawater over a period of time. And eventually, you'll stop being able to recover cells, live cells. But if you put a live squid in that seawater, the squid becomes colonized with the Vibrio fischeri. So the point is you can't detect them using traditional plate counting, but something about the live animal host allows the microbes to grow, obviously, in that natural context. So this is a really important concept, especially in terms of like food microbiology, where imagine you're a producer of some kind of food product and you're relying on viable counts in order to count live salmonella or whatever in your product. And if the salmonella go into a viable but non-culturable phase, you're not going to be able to detect them, and therefore you might erroneously think your food is safe and then distribute it and get people sick and get sued. And so these are the kinds of things that worry us and make us really interested in understanding what's happening in this viable but not culturable phase. And again, the term is a little bit misleading because clearly they are culturable. So in the case of the salmonella example that I gave you, you may not be able to detect the salmonella using traditional plate counting approaches, but if you ingest those viable but non-culturable salmonella, they can resuscitate in your gut and cause full-on disease. So that's why this is a really important topic and one that's gaining a lot of momentum in terms of physiological research. Programmed cell death is another hypothesis in death phase where a fraction of the population is genetically programmed to die, essentially. So it's sort of almost like an altruism. If 90% of your siblings crack open and their guts leak out, then the remaining 10% can live off those guts for a while and hopefully keep the population going when conditions get better. And so this GASP phenotype concept is a really neat physiological phenomenon. It's basically a loss of function allele. So RPOS is an important sigma factor that's essential for upregulating genes in stationary phase. Controlling genes in stationary phase. And so this RPOS is a interesting mutant deficient in this particular sigma factor, and so, or defective in this particular sigma factor. And so here's the amino acid sequence. So here's the normal sigma S, and the protein ends like this, LFRE. These are amino acids. And then the defective allele has this extra hunk of amino acids added to it, and that causes like I said, it causes a loss of function in the sigma factor. And so what's interesting here, though, is that, so again, you're looking in this graph, you're looking at log CFU per mil versus time. And so here's our RPOS mutant, the open boxes, and then the normal RPOS plus strain. And these are growing together in a culture medium over time. And what's interesting to note here is that the normal RPOS bacterium is unable to compete with the mutant. The mutant actually ends up outgrowing the wild type. For some reason, the loss of this stationary phase sigma factor gives this guy a competitive advantage. And so they referred to this as the GASP, growth advantage in stationary phase phenotype, from this particular science paper. And so some of the experiments that they did to show that there's this evolution of gradually improving strains of bacteria. Here's the RPOS GASP mutant competed, and basically you take a population of these guys and freeze them, and then the rest of them you grow up for about, you age for 2 weeks, you let them grow for 2 weeks, and then you compete these two against each other. So here's the parent mutant and then the progeny mutants grown together. And interestingly, the parent is unable to compete very well with the mutant. And you can do the same thing. You take these guys now and let them age, and then some you freeze, and then you take those guys out of the freezer and then compete them. And so here's the parent that you pull out of the freezer, and then the progeny mutants that you've aged for 2 weeks, and you throw those guys into a fresh flask. And again, the mutant progeny end up outcompeting the parentals in this particular case. And so there's this gradual positive selection for more and more hardy versions of the microbe. So it starts to speak to some interesting issues about what's happening in these death phases. It's not just if you go back to this original graph here, it's not just this kind of smooth decline. There's a lot happening underneath the surface here. And that's illustrated in this particular graph, where you see cell number over time. And what you're looking at when you're looking at optical density readings, or if you're looking at a plate counting, is just kind of a smooth line that suggests, a somewhat smooth line that suggests a less turbulent sort of decline in the population. But really what's happening, if you look at this genetically, is there's a bunch of successive evolutionary events, and new forms of the bacterium are taking over and then dying out, and they're undergoing these massive evolutionary steps and then extinction events in really short time. So this is the age of the dinosaurs right here, and then the age of the mammals and so forth, it all happening in a flask, all in record time. So how do we deal with viable but non-culturable in a lab setting? Well, one way to do it is to use live dead probes. Molecular probes makes a live dead stain that's really useful, and so the basis of it is there's a cyto9 dye and a PI, propydium iodide dye. And so imagine you have a live cell and a dead cell next to each other in a mixed culture. And so you would treat the cells with cyto9 dye. Cyto9 freely permeates into both live and dead cells, as indicated by the arrow. Propydium iodide, however, does not permeate into live cells, but strongly permeates into dead cells. And so as a result, the red propydium iodide floods inside these dead cells and stains them red, whereas the green cyto9 floods into the live cells and stains them green. Sometimes you get an intermediate, like an orangey looking cell, where there's a little bit of this and a little bit of that coming together, the green and the red coming together to make an orangey cell. But so you can imagine the cell's probably on its way out. But in this mixed consortium, you can see clearly red and green cells. And the live cells are predominant, and then some dead cells throughout. So it's a great way to give you a sense of what the population dynamics look like in a complex consortium. So this is a little bit about the stress response and things that happen that microbes do. Morphological changes are some of the biggest ones. Endospore formation is a real big morphological change, and another one is decreasing in cell size and shape. And the question then is, why would you want to shrink in size? And so the answer to that is purely mathematical. It can be answered in a purely mathematical way. The surface area to volume ratio changes as you get smaller. It actually increases. So if you're a big cell like this, your surface area to volume ratio is in this case 1.5. If you plug in the variables with your radius at 2 and so forth. And that's based on here. You look at the surface area and the volume calculations for a sphere. 4/3 pi r cubed as opposed to 4 pi r squared. So you have a cube function versus a square function. And so obviously things change dramatically as your volume changes. So in this little guy here, who has half the radius, or half the diameter, I guess, has a surface area to volume ratio of 3, meaning that this guy has a greater surface area to volume and therefore can bring in nutrients more efficiently and feed the cytoplasm of the cell more efficiently than this guy. And the problem is, obviously, we're talking about stress responses, and so the cells are probably stressed in some way. They're not they don't have the right amount of nutrients and so forth. So it behooves them to shrink up and improve their surface area to volume ratio. And obviously endospore formation is big because the spores are extremely resistant to environmental insult, and so they can therefore persist if they happen to be spore formers, which not a lot of organisms can do that, primarily a few types of gram-positive rod. And obviously there's a lot of production of stress proteins, RPOS and SOS proteins, which I'll talk about, and heat shock proteins and DNA binding proteins. And so these things will conspire to help the cell out. And so RPOS, I've already mentioned, is an a stationary phase sigma factor that associates with RNA polymerase and helps polymerase activate certain stationary phase genes. And the SOS response I'll talk about, but it's a pretty multi-pronged defense against a variety of stresses. And heat shock proteins, as the name suggests, can bind to DNA and protect them. And so again, these different types of activities are important. Some microbes will increase cross-linking in their cell walls to make them tighter and tougher, and many will protect their DNA with these DNA binding proteins, for example. Lots of different examples of chaperone proteins that can bind to proteins in DNA and so forth and protect them from degradation. And one of the things that microbes can often do is increase virulence, and that's something that we're keenly aware of as microbes become stressed. And that speaks to a lot of other issues too. So the hypervirulence of bacteria when they're in a biofilm state is as an example. So another version of this is the Corynebacterium diphtheriae that causes diphtheria, this sort of upper respiratory issue, and can also manifest as heart issues and so forth. But the bacterium is typically, when it gets into the body, experiences iron stress. And so the microbe may upregulate diphtheria toxin as a way to kill host cells to get access to this depletion of iron. The pH issues are pretty important as well. And most microbes, regardless of whether they're acidophiles or neutrophiles or alkalophiles, will maintain their internal pH near neutrality. The plasma membrane is impermeable to protons, and there's a lot of ion exchangers that are in place to facilitate a balanced pH. But there is an acid tolerance response, so they're capable of pumping protons out of the cell if they become excessive. And obviously that can be a problem because it does take energy to do that, but at least they're not dead. And so, and this is kind of the basis of a lot of preservatives that we use. We have a lot of examples of foods that are preserved with organic acids. They're not necessarily killing the microbes, but they're keeping them from growing because the microbes have to offset the increase in protons by pumping them out, and that takes energy, which again takes energy away from growing. Some synthesize acid and heat shock proteins that protect other proteins, and so again, or DNA. But there's another thing that happens too. Many microbes change the pH of their habitat by producing acid or basic waste products. And we've all seen this in different kinds of media that you've used, like the fermentation media that we use in 224 and so forth, or mannitol salt agar when you're growing Staph aureus and you see those yellow zones. That's the microbe is basically producing acid from the fermentation of sugar and acidifying its medium. But if they run out of sugar to ferment, they may do the opposite, where they start eating amino acids and pumping out ammonia and basic waste products. So they can modify their environment depending on they can switch metabolism to modify their environment. A good example of that is H. pylori, Helicobacter pylori, which is again a helical-shaped microbe with a tuft of polar flagella. So these guys could be ingested into the stomach lumen, and then they can penetrate their way through the mucus because they produce a variety of enzymes, and they have flagella that allow them to coil helically rotate and move towards the surface of a gastric epithelial cell. So in this particular way, they can, again, use flagella and chemotaxis to get where they need to go. But they also produce an enzyme called urease. And urease, in this particular case, will take urea that these cells naturally produce. And again, urea from the bloodstream and so forth comes and gets pumped out in here. But they utilize that urea and break it down into ammonia and carbon dioxide. And so the production of ammonia creates essentially a cloud of alkalinity around the cells, so they can actually survive in this type of pH environment, extremely inhospitable pH environment, by using their urease enzyme. Once they touch down on a cell, they can use a type 4 secretion apparatus, type 4 secretion system, to pump in toxins like CAG-A, which activates the production of the hormone gastrin. And so, and then they also produce a toxin called VAC-A, a vacuolating cytotoxin that you see here. It punches its way in the membrane and damages the gastric epithelial cells. But this combination of events producing this toxin and this toxin activates the production of gastrin, and it also messes with the proton pumps. And so you can end up with a situation like this, for example, hypergastrinemia, which is an excess of this hormone, and hypochlorhydria, which again is a reduction in the production of hydrochloric acid. And so an increase in the local pH here. And this combination of things can cause gastric cancers. So this is an interesting organism that's has a really neat physiology and is really capable of modifying its environment to adapt to this very inhospitable location. So how do microbes deal with oxidative stress? And again, oxidative stress from peroxide and superoxide radicals and all these things. Well, they use a couple different things, one of which is this transcription factor, OxyR. And these are sulfhydryl groups that you see here. So imagine this organism finds itself in the presence of peroxide, and these things become oxidized. In this state of oxidation, there's ultimately a disulfide linkage will be formed that changes the shape of this guy and allows it to activate the production of catalase, for example, and peroxidase, these types of enzymes that can break down hydrogen peroxide. So the effects of the peroxide on the transcription factor protein alter it to the point where it can now bind to a promoter to activate catalase and peroxidase, and therefore can break down the peroxide that stimulated its activity. Reactive oxygen species induced SOX-R dimers are also present in a lot of bacteria, and so in this kind of a oxidative environment, you see the oxidized SOX-R dimer, and once that happens, once this guy becomes oxidized, it can now recognize its promoter, and in this particular case, will activate SOX-R, as you see here, will activate SOX-S, and then SOX-S will bind to, for example, the superoxide dismutase promoter. And so superoxide dismutase can then break down superoxide radicals, which are included in these redox active compound group. And so superoxide dismutase will turn superoxide into peroxide. And then peroxide, of course, has to then be broken down into water and oxygen. Another example of this, and you're kind of zooming out a little bit so you can see a whole cascade of events in the presence of stress. So here's the no stress situation. These are happy flagellated bacteria swimming around in wherever, and inside the cell you're seeing a few things happen. The first thing is this MQSR, MQSA mini operon here, and so MQSA gets produced and will sit on its own promoter here and acts as an activator. But the main thing to note is that MQSA, when it's synthesized, its primary job is to act as a repressor for a variety of these stress response type genes, such as catalase and curli and cellulose. These are needed for biofilm. So curli is for attaching and cellulose is for the actual substance for the biofilm. And so again, the microbes there's no stress. The microbes want to be swimming and flagellated, so they don't need these sedentary activities. Cyclic diGMP is needed to activate these, and so you don't need that either in this circumstance. Not surprising, the flagella operons are allowed to be expressed under these conditions. MQSR binds to MQSA in this particular case too, and can keep it from doing, backing up and creating an overactive situation here. So this is important to keep this kind of keep MQSA in check. So some of the MQSA gets bound out and swamped out by MQSR. Some of it can do its job and cause repression. Now in the stress response situation over here, MQSA is again, in this particular case, MQSA is acting as the transcriptional activator here. MQSA under stress is now being broken down by this protease called LAN. And so RPO and again, MQSA, one of its jobs is to block RPOS. And so in this particular case, so in the presence of oxidative stress, for example, this protease becomes active, chews up the MQSA, as you see here, and breaks it down, freeing up this promoter. So as you can see here, MQSA blocks RPOS. That's what these flat lines are. That also, getting rid of MQSA, allows MQSR to become free. And MQSR is an RNA nuclease, and so MQSR can eat up any messenger RNAs that, for example, MQSA messenger RNA that get expressed. So that RPOS can now do its job, get synthesized, and block the flagella operon and activate these stationary phase essential stress responses like CSGD, which is needed for curli and cellulose. and the diguanylate cyclase, which is the signal that allows these guys to function optimally and to activate the catalase genes for combating the oxidative stress. Another thing that happens here is also in the stress response is the SOS response. And so in this SOS system here, the system is off because the LEX-A repressor binds to target genes. However, a small amount of REC-A is always made. And so here's our REC-A, and these are some of these genes, SOL-A, UMUDC, UVRA. And so LEX-A is a repressor. And so there's in the absence of any stress, LEX-A is on as the default setting. And so LEX-A will shut all these stress responses down, which is important because you don't want aberrant stress responses. You don't need a huge immune response, for example, if you're not infected by something. And so you want to keep everything damped down. However, a little bit of, as it says here, a little bit of REC-A is always made, because if you just shut things off completely, you could never activate it. And so it's got to be on a little bit. And so the REC-A protein is tiny little bits of it are made. And so in the presence of different indicators of stress, such as single-stranded DNA is a good indicator of stress because it suggests that the DNA is being shredded by something. And that's an example. And so REC-A, when it combines with a bit of shredded single-stranded DNA, sets off an alarm. REC-A becomes a protease, essentially, and can eat LEX-A, break it up. So in this combination, breaks it up. And now the repressor, LEX-A repressor, will fall off these promoters and allow them to do their job. So in this particular case, UVRA, for example, is part of the nuclear excision repair pathway. And so this means that it can fix mutations. And UMUD and UMUC, these are error-prone polymerases that can replicate across banged-up DNA, DNA with lesions. So these guys are active in times when there's lots of damage to the DNA. They can still work. And then SOL-A, for example, is an inhibitor of FTSZ, which should make sense, right? The microbe is under attack, is under stress, and doesn't want to be reproducing and wasting energy on that kind of thing. So inhibiting FTSZ in turn inhibits the cell division process. And so again, hopefully you can see that mechanism at play here, how REC-A looks for indicators of stress response and then becomes a protease to knock out LEX-A to free up promoters that are responsible for saving the cell from stress. And that's, again, the example of the SOS response. And this is by no means the extent of the SOS response. There's far more to it, but this is just a simplified diagram of that. And so looking at the nuclear excision repair program that cells have, you can kind of see the how it all layers on. Here's UVRA, and it's a dimer of UVRA. And so again, there's a lesion, this little star here, and RNA polymerase has hit that and has stalled. And that triggers this MFD protein to bind, and then the UVRA dimers to sit on that complex. This complex, UVRA, dimer, and MFD, provoke a dimer of UVRB to bind. And then in this particular case, that's, again, lesion recognition and verification. So MFD can now go away, and now you've got this complex. There's a, excuse me, UVRB makes a bit of a cut, and then the main enzyme, and then UVRA goes away, and then UVRC comes in, and UVRC, again, is looking for these dimers of UVRB, makes a dual incision, as you can see there, cut these little scissors, and that allows this thing to pull away the single strand with the lesion on it using UVRD. So UVRC cuts, goes away, UVRD comes in and lifts this away, again, can break the phosphodiester backbone and lift this away, and then obviously the DNA polymerase can come in and resequence across that and remove the lesion. So this is the nuclear excision pathway relying on the UVR proteins primarily. Here's another version of DNA repair. This is so-called photoreactivation. This one right here is light independent. It doesn't need light. But this one right here is light dependent, meaning that in order for this pathway to work, you have to have a certain exposure to a certain wavelength of light. And so imagine this little pyramid. Here's the lesion. There's a photoreactivating enzyme that recognizes that. Typically, it's called photolyase, lyase, L-Y-A-S-E. So the photolyase recognizes this damage, and as long as there's the appropriate wavelength of light, will absorb that light, become active, and cut that lesion out, and then restore the original structure of the DNA.